Interphase. For most cells, the majority of their life is spent in interphase. It is during this time that the cell carries out most of its normal functions, such as growth and protein synthesis. Interphase has three stages called G1, S, and G2. In many tissues, such as the brain, most mature cells will remain in interphase throughout their lives. G1 and G2 stand for first gap and second gap. Interphase begins with G1. This is the long period of time following mitosis, but before DNA synthesis, which is the S phase. During this time, the cell grows and carries out protein synthesis and other cellular functions, except DNA replication. The chromosomes are all unreplicated. That is, each contains only one molecule of DNA. The chromatin is very diffused within the nucleus, and so the individual chromosomes are not visible. The nucleus is complete, bound by a membrane, and the nucleoli are clearly visible with their proper staining. Prophase. During this stage, the DNA begins to condense into discrete bodies called chromosomes. Near the end of prophase, the nuclear membrane breaks down. Look closely and you may be able to see that the chromosomes are double. Each of the two chromosome subunits is called a chromatid. Also during prophase, microtubules in the cytoplasm form spindle apparatus or spindle fibers. And some of these microtubules, known as kinetochore microtubules, attach to the centromere of the chromosome. Metaphase. In metaphase, the spindle fully develops and the chromosomes align at the metaphase plate, a plane that is equally distant from the two spindle poles. Also, in metaphase, the spindle fiber attaches to the centromere of the chromosomes. Anaphase. In anaphase, the paired chromosomes or the sister chromatids separate and begin moving to opposite ends or the opposite poles of the cell. Spindle fibers not connected to chromatids lengthen and elongate the cell. At the end of the anaphase, each pole contains a complete compilation of chromosomes. So it's like the spindle fibers tear apart the chromosomes and place them at the side, just like what is being shown in the picture. Telophase. In telophase, the chromosomes are cordoned off into distinct new nuclei in the emerging daughter cells. Also, in telophase, the nuclear membrane appears, the nucleolus disappears, and the spindles disappear. Cytokinesis. Cytokinesis is the division of the original cell cytoplasm. It begins prior to the end of mitosis and completes shortly after telophase. At the end of cytokinesis, two genetically identical daughter cells are produced. Thank you for watching this video on the stages of mitosis, and we hope you enjoyed. Bye!